Hello everyone. We've gotten to the point where this semester so far we created a vector that had an sgRNA sequence in it to be able to allow us to be able to make a mutation in yeast. Then we transformed using a repair template and that plasmid to be able to create a mutant. And the next following steps where we went ahead and grew up these cells. We isolated protein from them. We did a chromatography step, and in that step we collected a number of fractions. And then the following week we assayed each of those fractions for enzyme activity and for protein concentration. And now we're going to the final experiment of the semester, and we're going to do this 2 weeks in a row, and just to be able to reproduce our data. And this is, we will go ahead and we'll determine some of the enzyme kinetic parameters for our mutant enzyme, and we'll actually assay the wild type enzyme at the same time so we can make direct comparisons about activity. Let's go ahead and get started on that. And the first thing that I think we should do for this is we need to make standard for, we don't know how much material we made. The product of the reaction is we're adding alkaline phosphatase and we're using phosphonitrophenyl phosphate as a substrate. And that substrate is a clear solution. This one's slightly yellow, but it's a clear solution. And then whenever the enzyme acts on it, it becomes a yellow solution. So you can see here maybe a little bit of product has been made in this sample already, and we'll go ahead and start that. So our first thing to do is, this is the yellow sample. This is 500 millimolar PNP. This is phosphonitrophosphates. It's commonly used in a lot of these kind of assays. And what we'll do at first is we'll add 700 microliters of our assay buffer to each of several tubes. So I'll go ahead and do that right here real quick. So what I have here are 4 tubes labeled 250 micromolar, 125 micromolar, 62.5 micromolar, and 31.25 micromolar. And I've added 1 milliliter, 7/10 of a milliliter of the assay buffer, which is 20 millimolar tris pH 9 with 20 millimolar magnesium chloride present. The next step is to go ahead and do our serial dilutions. And from there, I will take 700 microliters of this solution. That's 500 micromolar, or 0.5 millimolar, and I will add it to the tube that I have labeled 250. I will mix it by pipetting up and down. I will take 700 microliters out of this tube, close it, and I will add it to the tube labeled 125. Take 700 microliters out and add that to 62.5. Mix that up, and I'll take 700 microliters out and add it to our last tube, in which I will do the same thing yet again. This one we don't have to take out because it's the last tube. So now we have the first part set up where I now have 4 tubes at different concentrations of PNP. And I have the more concentrated one. I'll go ahead and put this right alongside it. So there's the 5 samples set here. The next step is to go ahead and we need to make our substrate. And unfortunately, we kind of have to do this twice because of volume constraints. But I will go ahead and add one milliliter to each of 10 tubes. So what I've done so far is I have these tubes already labeled. The first tube here is labeled 10 millimolar. The second is 5, 2.5, 1.25, and 0.625. And I have a duplicate set of tubes labeled in identical fashion. And notice that I have them lined up in terms of decreasing concentration. That makes it a little bit easier and helps prevent errors. So now what I'm going to do is I'm going to add 1 milliliter to the tube labeled 10 millimolar. I will pipette it up and down to mix it. Being very careful now because you're to the very top of the volume of this tube. So be a little careful when you're doing this mixing. And now I'm going to take 1 milliliter out, cap that tube, and I will add this to the tube labeled 5 millimolar. And just be careful doing this because these are 2 mil tubes and then we're adding 2 mils to them. So I'll take a milliliter out of the 5 millimolar and add it to the 2.5 millimolar. To the 1.25 millimolar. And then we'll add it to the 0.625 millimolar. Here we have 2 milliliters. So what I'm going to do here is go ahead and repeat the same process again for another set of tubes. Take 1 milliliter out of the stock solution, add it to the first tube, the one that I have labeled 10 millimolar. I've done this a couple of times this semester, so I'll just quickly do this real quick. Go through here and do this and get to the end. Okay, what I've done is I've done the serial dilution twice, and we're going to be using, the reason I did this twice is we're going to need about 1.5 milliliters, and we don't have any tubes that are about 3 to 4 milliliters, so I decided to go ahead and have you guys do it twice. 1, to practice, and 2, just for practical reasons. So what I'm going to do now is I'm going to open up all these tubes. I don't need to add the last one because there's 2 milliliters in here. And then what I'll do here is I'm going to transfer the contents of this tube into this tube. So what I now have is a series of tubes approximately 2 milliliters of 10, 5, 2.5, 1.25 millimolar, and 62.5 millimolar PNPP, which is our substrate for the reaction. So we have our standards for the standard curve over on this side, and now we have all of the materials we need to be able to do the reactions to do the enzyme kinetic study. Looks like my enzyme is not quite thawed out, so what I'll do is I'll go ahead and stop now, and we'll wait for a few minutes till this tube is thawed, and then we'll set up our plate. I'll be back to you in a few minutes. Bye. Okay, everyone. The enzyme sample I have, both of them have now thawed out. I'll be using the same one twice because remember I only did I just did the wild type. I didn't do a mutant. So I'll treat this wild type enzyme as a mutant as well. You will have 2 enzyme solutions, and the reason I chose this one, this is the 0.3 molar, the first fraction. It was the one that had, of all the column fractions, it had the most activity. It turned the yellow the fastest, and it had the most enzyme activity, and in about 20-minute assay, I was able to get a significant amount of yellow, so I figured this would be the best one to use. So I'm now making sure it's mixed well. Notice I have the bubble looking it back and forth, making sure it's nice and mixed. So I'll set it off to the side here. So we're going to lay it out on the plate, and what I kind of like to do is talk about sort of what I think about when laying out an assay on a plate. For example here, what we're going to do is we're going to have the first 3 rows here are going to be PNP standards. Okay, so all of these will be PNP standards. So we'll know this part of the plate we use to make the standard curve, determine the amount of product produced. This row D is going to be sort of a control to see at what rate does the PNP form just because it's in buffer. I'm not sure. I noticed that there was a little bit of yellow here in the buffer, in the substrate, so I want to go ahead and do that. And so what we'll do is we'll subtract these values from the enzyme activity values we have in these wells. So these wells will have 20, 10, 5, 2.5, 1.25, and 0 millimolar substrate. And the first 3 rows here will all be wild type enzyme, and the next 3 rows down will have the mutant enzyme. So the plate will have 4 sections: the PNP standard, sort of the background control for product formation just in buffer. I'm not sure if this will give us a significant amount of information or not. I haven't done the assay enough to really know, because I've been told that it sometimes happens. And we have the enzyme kinetic samples, 20, 10, 5, 2.5, 1.25 across here. We have wild type and mutant enzyme here. Okay, so let's go ahead and get started. So in all these wells here, you'll notice that I laid out things in kind of in an order and laid them out. So right here, in row 7, A, I'll put 200 microliters of substrate of the 20 millimolar. I'll put that into 6 wells going down. It's all this 7A, B7, C7, and so on. Okay, so we now have 6 samples loaded of the 20 millimolar substrate. So this is the 20 millimolar substrate section. Next, we'll do 10 millimolar, which is the first tube from our serial dilution. And what I'll do is I'll lay these out on the plate. You can do these in any particular order that you want. I just go ahead and generally try to do all the columns and the rows first. It looks like I made one extra dilution that I didn't need to make. That's okay. And next is to put the blank all the way down in column 12. Okay, so let's go ahead and go back through what I've got accomplished so far. We have 20 millimolar PNP in column 7, rows A through F. And in 8 we have 10 millimolar. In 9 we have 5 millimolar. In 10 we have 2.5. In 11 we have 1.25. And in 12 we have just a buffer blank. So we have from a high concentration down to lower, lower, and lower concentration. And so the next thing to do is we'll need to add enzyme to all of these. And we'll add the same amount of enzyme to every one of these. So it'll be 20 microliters. And you guys will be putting, the first 3 rows, you'll be putting the wild type enzyme. And the next 3 rows, you'll be putting your mutant enzyme. I don't have a mutant enzyme right now, so what I'll do is I'll just go ahead and go across. I'll just put the same amount in there. So check to make sure your pipette is at 20. We're actually isolating very little enzyme, and in this case, that's why we're having to do reactions that are 20 minutes long. Normally, if you have a lot of enzyme activity, you'd be doing these over maybe 5 minutes, and in 5 minutes, you'd have to be more careful about making sure the enzyme and substrate work together for exactly the same length of time and doing a timed reaction. But unfortunately, or maybe fortunately for this, we don't have the enzyme concentrations aren't high enough, and we're not seeing that rapid of a reaction, so they're pretty slow. So now I've gone through and I've added enzyme to all of these wells. The next step is to go ahead and create our standard curve. And those, we use these tubes here. And here I'll go ahead and set up the pipette to 220 microliters. In this case, I'm using a P1000, setting it to 220 microliters. And I'll put those in columns A through C. Column 1 through C. Oops. So column A. And this is the 20 millimolar here, the 500 micromolar here. In column 1. Change the tip. So here I have the 250 micromolar, sorry. In C, in column 3 will be 125 micromolar. Column 4 is 62.5 micromolar. And in column 5, we will have the 31.25. And for a blank, we'll go ahead and add buffer in for our blanks. It's the same buffer with a substrate in it. Put those in column 6. I know your protocol doesn't mention it, but I'm going to go ahead and put the blank in column 6 in row D as well. Now, I made a comment that I wanted to go ahead and create a control of just the substrate incubating the same length of time to see if there's any autohydrolysis or anything like that that happens. So I'll be adding the standard in these wells. So we have the standard here. Oops. Okay, we'll add the 20 millimolar in E. Notice I'm adding 220 microliters this time. In B is 10 millimolar. 2.5 millimolar in 4. 1.25 and 5, and there we have our standard curve and our hydrolysis. Now we just let these incubate for 20 minutes, but let's make sure, look at your plate and make sure that we have some yellow color forming in column 7 and 6 for sure, and then bring your plate to the TA or myself and we will read the plate. And I will generate a video on processing the data from this plate. And hopefully everything works well, and we'll see you in the lab.